Welcome everyone who have joined. We can just wait for 2-3 minutes. So welcome everyone. Let's start the session. I welcome you all to the week 2 live session for the functional genomics. Is my screen visible to all? Can someone confirm? Is my screen visible?
Yes. Yes.
Okay. So firstly, I like to give some questions to you based on the discussion which we had previously. So the very first question is, given here is a strategy to generate a knockout mice where the process is marked as Y, and this process helps in the generation of the knockout allele from the targeting vector and the wild type allele of the gene of interest that has to be knocked out. So the process labeled as Y here is what? Option A, homologous recombination. Option B, non-homologous end joining. Option C, crossing over. And option D, none of these. So anyone who would like to answer this question can either answer in the chat box or can unmute and answer. Does anyone like to answer or should I explain? The correct answer is homologous recombination. So this setup is basically for the removal of the target gene with knockout of the target gene with the neomycin resistant cassette. So in this, what happens is by the process of the homologous recombination, the neomycin resistant gene gets incorporated and our gene gets knockout, leading to the formation of the knockout allele. The next question is, given here is a pair of mouse having in its genome with indicated construct for creating a knockout mouse model. Which of the following options do you think correctly identifies the possible outcome of this cross? So this is a Cree-log speech system, and what will be the possible outcome if we cross these two mice? The first option is the target gene will be deleted in all the cells of the resulting mouse. The second option is the log-sp segments will be lost in the genome. Third option is the target gene will be deleted only in the neurons. And the fourth option is the Cre recombinase will express in the neurons. So anyone who would like to answer this? Okay. So the correct answer is the target gene will be deleted only in the neurons. This is because in this Cre-Loxp system, we have the Cre recombinase enzyme, which is downstream to the neuron specific promoter. So it will only be expressed in the neuron cells. It will only be expressed in the neurons, and therefore the target gene will only be deleted in the neurons. So this is a conditional knockout setup. Question number 3 is, which one of the following statements correctly differentiates between a transgenic animal and a knockout animal? Option A transgenic animal is used to model both loss of function and gain of function mutations, whereas a knockout animal can be used to model only the gain of function mutations. Second, a transgenic animal may have a foreign gene inserted in its germline, whereas a knockout animal has an endogenous gene that is inactivated or modified. Option C, a transgenic animal can be used to study diseases that occur later in life, whereas knockout animals can be used to study embryonic development. And option D is none of these. So anyone who wants to answer this question, you can write your answers in the chat box if you do not want to unmute. Okay. So the correct answer is, a transgenic animal may have a foreign gene inserted in its germline, whereas a knockout animal has an endogenous gene that is inactivated or modified. So anyone who knows any example of the transgenic organism or any transgenic animal which they know? So one such example is dolly sheep, which is a transgenic animal, and it is produced as a result of the foreign gene which is inserted in its germline. So coming to the week 2 module, what there are certain other genetic approaches. There are certain other approaches which are used for the gene lockdown. So the first approach is the CRISPR-Cas9 approach. CRISPR-Cas9 is a genome editing approach that actually exploits the natural DNA snipping enzyme, which is the Cas9 enzyme, that is CRISPR-associated 9 protein, to target and edit the required gene. So CRISPR stands for the clustered regularly interspaced short palindromic repeats. And these are naturally present in the prokaryotic system as a part of their immune defense. So and scientists have actually used this CRISPR-Cas9 technique for the gene knockdown approaches and the gene knockout approaches. So how CRISPR works? Basically, the Cas9 protein, it forms a complex with a guide RNA in a cell, and this guide RNA is a complementary sequence that identifies the target gene which we have to knock out. So this complex then attaches to the matching genomic DNA sequence which is adjacent to a spacer. Here it is the spacer which is represented by a yellow segment, and this guide RNA binds to the complementary sequence which is present in the genomic DNA. And due to the this Cas9 RNA complex and cuts the double strands of the DNA, and this leads to the insertion, deletion, or the frame shift in the required in the programmed DNA. So the insertion or deletion may occur as a part of the this double strand breaks which are created by the Cas9 RNA complex may be filled either by the process of non-homologous end joining, or they can be repaired by the homology directed repair. So in case of homology directed repair, in case of non-homologous end joining, there will be no involvement of the homologous flanks. But in case of the homology directed repair, there is a donor DNA which consists of the homologous arms as as the one which has undergone the DNA damage. So this donor DNA basically undergoes homologous recombination with the cleaved DNA, and the new DNA is inserted into the as a process of homology directed repair. So any doubts? Okay. So the next question is, which one of the following method need not always inactivate a gene function? Option 1, RNA interference. Option 2, CRISPR-Cas9 method. Option C, knockout mice. And option D, none of these. So anyone who would like to answer? RNA interference I will explain in details later. So anyone who would like to answer, what can be the possible, like any guess? What can be the possible answer? Okay, D. Can you explain why do you think none of these can be used to inactivate, will not always inactivate a gene function? So actually the correct answer should be the CRISPR-Cas9 method. As I told in the previous slide, that the new DNA can also be inserted as a part of the homology directed repair. Therefore, it is not necessary that in using CRISPR-Cas9 method, we'll always inactivate a gene function. We can also leads to the activation of some gene functions by incorporating the particular DNA fragment by using the homology directed repair method. Next question is, which one of the following steps is not involved in the CRISPR mediated technique of genome editing? First, formation of the Cas9 gRNA complex around the target gene. Second, activation of the double-stranded break repair machinery. Third, double-stranded breaks in the gene of interest by custom-designed endonucleases. And fourth is non-homologous end joining of the double-stranded breaks at the site of gene insertion and/or deletion.
Ma'am, option B.
Uh, okay. Uh, why?
Because ma'am, in the time of CRISPR-Cas9, if the double-stranded break repair machinery is activated, then we are not satisfied with our work means targeted DNA enter in the breakage point.
Okay. Augustine has wrote fourth option. Can you please explain? And can you please unmute yourself and explain this?
Because the joining is usually homologous end joining.
No, it is not like that. That it is usually, but it can be non-homologous end joining as well. So, I was asking which of the following steps is not at all involved in the CRISPR-mediated technique of genome editing. So, the correct answer is that the double-stranded breaks in the genome of interest is not done by the custom designed endonucleases, as the endonuclease which is used in the CRISPR is, we all know, is the Cas9. So it is not the custom designed endonuclease as the endonucleases which are used in the other genome editing approaches. So for SK Murtaj, who said that The option B is correct, that activation of the double-stranded break repair machinery does not take place. So the last step of the CRISPR-Cas mechanism should definitely be the repairing of this double-stranded break which we have created as a part of the this CRISPR-Cas9 technology. Also, I have explained why fourth option is not correct. I hope it is clear to all.
Yes, ma'am.
Okay.
Yes.
So the next question is, identify the name of the enzyme which is used in the CRISPR-Cas9 genome editing approach. It is very easy. It is. So what is the enzyme which is used? Caspase 9, Cas9, Crispase or Cris9?
And Cas9.
Okay. So it is a theoretical question which most of you know, that the enzyme which is used in the CRISPR-Cas9 technology is the Cas9 enzyme. Next question is, given here is a schematic of a method for the genome editing with engineered endonucleases. Here, the specificity of the restriction endonuclease to cut at the gene of interest, which is identified, which is here shown marked by the red arrow, is guided by which of the following? Option A protein which is localized on the gene of interest. Second, a complementary sequence to that of the gene of interest being present at a part of endonuclease complex. Option C, an antibody anchors the endonuclease to the gene of interest. And option D, modified histones near the gene of interest show higher affinity for the endonucleases. So the question is clear to all?
No, ma'am.
Okay. So here in this diagram, what I am depicting is, this is the schematic of a method for genome editing approach using the engineered endonucleases. So endonuclease, as we all know, will cut in the DNA. And here, how will the restriction endonuclease will cut at the specific site, which is our gene of interest? So what is the property that should be there in the as a part of the that provides the specificity to the restriction endonuclease to cut at the particular site, which is the uh, site of our gene of interest? So is it something, a protein which is localized on the gene of interest, that is helping in identifying the restriction endonuclease enzyme? Or it is a complementary sequence to that of the gene of interest being present as the part of endonuclease complex? Or is it some antibody which is anchored to the endonuclease as a part of endonuclease to the gene of interest? Or there is some modification in the histone proteins near the gene of interest and therefore providing the higher affinity for the endonucleases?
Ma'am, option B.
Okay. Divya, would you like to unmute yourself and answer, since you are answering in the chat box? Okay. So yes, the correct answer is option number B. So a complementary sequence, as we see in case of CRISPR-Cas9 technology also, where the guide RNA is the complementary to the our gene of interest, which we are uh, where the endonuclease will work. So similarly, in this case, where we are performing genome editing with engineered endonucleases, the endonuclease complex consists of a complementary sequence which is complementary to the gene of interest, and this sequence will come and recognize the gene of interest and will bind to the gene of interest, and as a result the endonuclease activity will take place, and therefore the endonucleases, which are also known as the molecular scissors, will cut at the specific site. The next question is, shown below is a schematic. Next question is, shown below is a schematic of a technique used to engineer the genome to understand a disease process or gene function. Which one of the options given below correctly identifies the property of non-homologous end joining and homologous recombination indicated by the red color arrows in the schematic? So first option is that they repair the double strand break which is created as a part of the endonuclease activity. Second is they creates double stranded DNA break. Option C, they insert new double stranded DNA breaks, and option D is none of these. So what is the function or what is the property of this non-homologous end joining and homologous recombination or homologous directed repair which is taking place?
Option A, they repair the double-stranded DNA break.
Okay. So it's again a theoretical question, and the answer is it repairs a double-stranded DNA break, which is created as a result of the endonuclease activity. The next concept, which was introduced by Professor Ganesh, was the central dogma of molecular biology. So the classical concept of central dogma of molecular biology states that the DNA gets transcribed into the RNA by the process of transcription. This RNA then, by the process of translation, leads to the formation of the protein. But the modern view of central dogma of biology has some additional steps to it, where it says that not only DNA gets transcribed into the RNA, but DNA also undergoes the self-replication with the help of the enzyme DNA-dependent DNA polymerases. Similarly, RNA might undergo self-replication with the help of RNA-dependent RNA polymerases or the replicase enzyme, which are present in certain viruses that helps in the which are present in certain retroviruses or the RNA virus that helps in the replication of the RNA in those viruses. And this RNA leads to the formation of the protein, and there are certain proteins which are self-propagating. For example, one of them we know is the prion proteins, which are present in the scrappy form in the disease condition. So this is the modern concept of. Also, one more step which I missed is that the RNA may get reverse transcribed into the DNA with the help of the enzyme which is reverse transcriptase, and the process is known as the reverse transcription. The enzyme which is responsible for this process is reverse transcriptase or the RNA dependent DNA polymerases. So there are in our genome not only coding DNAs are present, but there are certain non-coding RNAs which are also present. So this figure A here shows that the non-coding RNAs comprises almost 99% of the primary RNA transcripts that are generated by the genomic transcription. Beside this, the non-coding RNAs comprises 80 to 90% of the poly-A tailed mature RNAs. Since the mature RNAs have a poly-A, that is polyadenylated tail, so this non-coding RNAs also have this, some of these non-coding RNAs also have this poly-A tail, and they comprises of 80 to 90% of the mature RNAs. Over 60% of the protein coding genes, these non-coding RNAs may include the miRNAs or the long non-coding RNAs or small nuclear RNAs. And the 60% of the targeted protein coding genes in humans are targets of the miRNA, which is produced produced inside the human cells. About 70% of the protein coding genes have at least one homologous antisense for the non-coding RNAs. So the next question is related to central dogma. The image shown here is a classical view of the central dogma of molecular biology, and the different steps are marked with the numbers 1, 2, 3, and 4. So which of the following options correctly identify the step with the correct name of the process?
Now option D.
Okay. So rightly said that option D is correct. Uh, Here the number one shows the replication of the DNA. Number two here shows the DNA gets transcribed into the RNA by the process of transcription. Third is the RNA getting translated into the protein, and the fourth process is the reverse transcription, where RNA gets converted into the cDNA. The next question. The central dogma of molecular biology is an explanation of the flow of genetic information within a biological system. Which one of the following options does not fit into the classical concept of the central dogma? Option A, protein. Option
Option
B,
B.
okay. Anyone else who would like to answer? Okay, the correct answer is the non-coding RNAs, as the classical concept of the central dogma talks about the DNA gets transcribed into the mRNA, which are the coding RNAs, and this messenger RNA then translates into the protein. So the central dogma of molecular biology does not talk about the non-coding RNAs. How is it possible that a certain genes leads to the formation of multiples of mRNAs? So the process which is involved in this is the alternative splicing. In this figure, we can see that this is the nuclear RNA, which consists of the potential exons 1, 2, 3, 4, 5, which are separated by the intronic sequences. So this, as a part of the alternative splicing, what may happen is that this nuclear RNA may undergo splicing in the form that the exon 1, 3, and 5 may leads to the formation of the mRNA, or it may select some other exon, that is 1, 2, 4, or 5 may be selected in another mRNA, leading to the formation of different mRNAs from a single nuclear RNA. So, this is an example that how alternative splicing plays an important role. This alternative splicing is is from the family of a red alpha tropomyosin proteins. The alpha tropomyosin gene is presented here on the top, and the thin lines here represents the sequences that becomes introns and are spliced out when the formation during the formation of the mature mRNAs. Constitutive exons, which are found in all tropomyosins, here are shown in the green. Those expressed only in the smooth muscles are shown here in the red, while in others, this exon is not selected. And those which are expressed exclusively in the striated muscles are shown with the purple. So those that are variably expressed are shown in the yellow, and these are expressed in the different like striated muscles 1, 2, and myoblast. And in addition to many possible combinations or exons, two different 3' ends and the 5' ends are therefore uh, possible as a part of the alternative splicing. So is it clear? So in short, we can say that alternative mRNA splicing is a mean of producing a wide variety of proteins or a wide variety of mRNAs from the same gene, depending which exon is being selected as a part of the mRNA. And so based on this, we have a question. Alternative splicing is not a process in which the newly formed mRNA is differential spliced. Which one of the following options is not an expected outcome of the alternative splicing process? Option A, it leads to the formation of 5' alternative 5' end or 3' end of the mRNA. Second, it generates distinct polypeptides from the same gene. Option C, differentially spliced mRNAs are the major contributors to the non-coding RNAs in the cell. And option D, alternative splicing shows tissue specificity. So anyone who would like to answer? Okay, option number 3. Hmm. Ricky, would you like to explain this?
Can I see the question once? Because it's
Yeah, yeah, yeah,
blackout.
yeah.
Because spliced mRNAs are the ones who doesn't contain any intronic region, whereas during the process of splicing, the introns are getting spliced out, and which later on introns are acting as a microRNAs, which halts the translation. So the non-coding RNAs in the cell are not linked to the spliced or the matured mRNA.
Right. So the correct answer is the differentially spliced mRNAs are the major contributors to the non-coding RNAs in the cell is the wrong option. Uh, the next question is something based on the calculation. So this is the DSCAM gene of Drosophila that contains 24 exons. Out of these 24 exons, exon 4, 6, 9, and 17 are encoded by a set of mutually exclusive possible sequences. Each messenger RNA will contain one of the 12 possible exon 4 sequences. Similarly, one of the 48 possible exon 6 alternatives, one of the 33 possible exon 9 alternatives, and one of the 2 possible exon 17 sequences. The Drosophila discam gene is homologous to a DNA sequence on human chromosome 21 and is expressed in the nervous system. Disturbances in this gene may lead to the Down syndrome. So, this is for the general information. I want you to calculate the total number of proteins which you think can be encoded by this gene.
And 3672.
Okay. Can you explain how did you calculate this?
Because ma'am, here we got the exon number, exon 4, exon 6, and exon 9, and exon 17. Then we get here 4 region of exon 4, and
Yeah.
then exon 6 also 6, exon 9 also 9, and exon 17 also 17. Now, when the alternative splicing or exon shuffling occurs, then the every gene rearrange with each other another dependently. So here
Okay.
the probability of the outcome of the gene is multiply this number of
Number of
gene.
alternatives for the each exon, right?
Yes.
Okay. So, it can produce approximately 38,000 theoretically 38,016 different types of protein by alternative nuclear RNA splicing. Any doubts, or is it clear to all? Okay. So let's move to the next concept, which is, yes, okay, not clear. So, okay, let me explain it again. So this is the D-SCAM nuclear RNA, which has a 24 exons. Okay. Out of these, exon 4, exon 9, exon 6, exon 9, and exon 17 have different alternatives. Like exon 4 has 12 alternatives, out of which only one will be taken up in the mRNA, which is the mature mRNA, which is the mature RNA, or the messenger will be taken in the messenger RNA. Similarly, out of the 48 alternatives for the exon 6, only one will be picked up in the mRNA. And for the exon 9 and exon 17, the same case will happen. Out of 33 alternatives for the exon 9, one will be picked up in the mRNA. And for exon 17, out of two alternatives, any one of these two alternatives will be picked up in the mRNA. So we need to calculate here. We need to calculate that how many number of combinations are possible here. So when we see that out of 12 alternatives, one can come. So this total number of mRNAs which will be possible as a part of mutually exclusive possible sequences will be the multiplication of these 12 into 48 into 33 into 2. That is 38016 different types of mRNA translating into the 38016 different types of proteins. Is it clear now?
Ma'am, what is the DSCAM nRNA?
Nuclear RNA, which is not imported into the cytoplasm.
Yes, ma'am.
DNA first,
Nuclear RNA convert to mRNA.
yeah,
So the protein is generated from mRNA.
yes,
So ma'am, here actually the mRNA contain how many types of exon 4 in nRNA
only
exon
one,
4?
only one. mRNA
Yes.
will have only one out of these 12 alternatives, only one alternative will be present in the mRNA.
Okay,
Similarly,
okay,
for
got it.
exon 6, only one alternative will be present in the mRNA. Is it clear?
Yes, ma'am, got it.
Okay. So let's move to the next concept of RNA interference. With the discovery of RNA interference, specific gene knockout knockdown tools actually have become available to the scientific community and are developed towards the clinical applications. Here, in general, a gene-specific knockdown can be achieved in mammals through the application of the antisense RNAs, antisense which is complementary to the RNA which we are trying to interfere with, and we are trying to knock down, and these miRNAs are the endogenous RNAs, which are the non-coding RNAs, as I explained earlier, and some exogenous small RNAs, which is the small interfering RNAs or the siRNAs, are also available, which is the small inhibiting RNAs. And this siRNA and miRNA leads to the process of the RNA interference, leading to mRNA decay and the translation inhibition. So what happens is the miRNA gene which is present leads to the formation of the primary mRNA molecule, which is a hairpin loop-like structure. This Trosha molecules then binds to this hairpin structure, leading to its cleavage and leading to the formation of the pre-miRNA. This pre-miRNA then is exported from the nucleus to the cytoplasm, where it interacts with the dicer molecule. This dicer molecules cleaves this hairpin structure and leading to the formation of the miRNA duplex. These miRNA duplex unwind from each other, and the asymmetric strand of this miRNA is loaded into the RISC, which is the RNA-induced silencing complex. This RNA-induced silencing complex then may either interact partially with the coding regions of the mRNA, thus leading to the translation repression, or may interact with the target mRNA uh, in a completely with the target RNA, leading to the mRNA cleavage. So, is it clear?
Yes, ma'am.
Okay. Gene knock silencing, same regarding modulation of gene expression. So there is a difference between gene knockdown and gene silencing. So basically, in case of, sorry, are you asking about gene silencing or gene knockout? Yeah, this is similar with respect to if we are talking about the gene expression. But if we are talking about the gene knockout and gene knockdown, then there is a difference because in gene knockdown, what we do is we just decreases the uh, protein translation, but will not completely, the gene is still there in the genome. But in case of gene knockout, what we do is we remove the gene of interest from the genome itself, and then we check for the function of the gene. So, how we deliver this siRNA or miRNA into the cells? Firstly, this siRNA or miRNA have to pass the endothelium and penetrate the cell membrane to become incorporated into the RISC complex. So, there are certain potential we can say that there are certain potential barriers, for example, nucleases that may act on these miRNAs or the siRNAs and therefore can degrade them, or therefore resulting in the inefficient target knockdown. So there are different delivery strategies which are adopted for the delivery of the siRNA or the miRNA for the gene knockdown purposes. So we can either directly deliver the siRNA or miRNA, but there is a disadvantage of rapid degradation, accumulation in kidneys, and immune stimulation. We can conjugate this siRNA and miRNA with certain antibodies. Cholesterol conjugation may takes place. Aptamer, we can use the aptamer for the conjugation, or we can use the liposomal encapsulation. So these different methods actually offer certain advantages, therefore preventing the rapid degradation of the siRNA and miRNA and leading to the increase in the efficiency of the gene knockdown. So the schematic given below here represents which of the following tools of the functional genomics. Option A, RNA interference. Option B, Krylov-Sp system. Option C, homologous recombination. And option D, morpholino oligos.
RNAi.
Okay. Do you know what is morpholinos? Oligos.
No, ma'am.
Okay. So why do you, okay. Can you explain the process here described in the schematic? What is happening? Why do you think it is RNA interference?
Because, ma'am, when the mRNA is transcribed from DNA,
Okay.
then the complementary RNA is come in and bind with the mRNA
Okay.
and inactivate the expressible mRNA and also cut by this expressible mRNA with other helps of enzyme.
Right. Okay. So anyone who knows what are morpholinos? Okay, so these are also the oligomers, which are oligomers. We can say that they are similar to the DNA and RNA sequences, but the difference is that they contain a morphylin rings. So they are also used in the molecular biology to modify the expression of the genes, and its molecular structure contains the DNA bases which are attached to a backbone of the methylene morpholin rings, and hence the name morpholinos. And they linked through the instead of phosphodiester bond, they are linked through a phosphorodiamidate bonds, and the morpholinos block the access of the other molecules to the small with other molecules that are to the sorry, these morpholinos actually represents the excess of the other small molecules with the specific sequences of the base pairing surfaces of the RNA. Thereby the RNA will not be able to translate and therefore will not be able to produce the protein. So this is basically a approach which we can use as a tool for the reverse genetics if we want to study by knocking down the gene function. The next question is some based on some research activity. So a group of students was given the assignment to construct a library of genes that could possibly play a role of functional role in mitochondrial function. So we have to prepare a library of genes that play a role in mitochondrial function. Which one of the following correctly identifies the correct sequence of the proposed study design? Like what are the steps we should follow? Like what is the sequence of the steps which we should follow to create this library of the genes? Anyone who would like to answer? You can take time because it is a lengthy question. So the correct answer is firstly, we will establish a stable cell culture system with fluorescently tagged transgenes to permit live visualization of mitochondria. Next, we will select for the genes which we have to for which we have to perform the gene knockdown. So we will next do the bioinformatic selection of the candidate genes for the knockdown. Next step will be the designing of the RNA interference based screening to knock down the transgenes which we have selected using this bioinformatics various bioinformatic tools. The next step will be the when we interfere the RNA, that is when we perform the RNA interference and we knock down the transgenes, then we will try to visualize. So what happens in the mitochondria? So next step will be the visualization of mitochondria in cells transiently transfected with the specific siRNA. And the last step will be definitely the data analysis and compilation in the database. So this will therefore help in constructing a library of the genes that may possibly play a functional role in the mitochondrial function. Any doubts? Or if it is not clear?
Yeah, ma'am.
Okay. Answer is mentioned here only. So mitrons helps in the regulation of gene function in a cell. What are the mitrons? So since the answer is mentioned, so mitrons are miRNA, that is the microRNAs that is derived from the spliced introns in the non-mammalian species. As someone explained it earlier as well, that the introns may spliced introns may sometimes act as a miRNA, but these mitrons are not present in the mammalian species. They are present in the non-mammalian species like the C. elegans and Drosophila melanogaster. The next technique, which is widely used in studying the transcriptomics, is the microarray. So, microarray permits the analysis of the gene expression and DNA sequence variation, protein levels which are present in the tissues or cells, and other biological and chemical molecules in a massively parallel format. The basic concept of the microarray is the hybridization, in which our gene of where we either try to hybridize our gene of interest or the mRNA with the complementary DNA sequences which are fluorescently labeled. And hybridization will only occur when there will be the complementarity. So, therefore, by using the different probes, by using the known probe against a set of genes, we can see that whether which gene it is binding or to which mRNA it is binding, therefore producing the required fluorescence. So this is the basic schematic diagram where we purify the mRNA. So this is the basic schematic diagram where we purify the mRNA from the cells, we label them, and then and produce the complementary DNA. We mix them, hybridize them, and wash them, and then we scan them using the laser. So the usually what color scheme is used for the microarray is the green here shows the activated gene, red here shows the repressed gene, and the ones which are neither activated nor repressed here is shown by the yellow color. So, or the other schemes which can be used is the overexpression can be shown by the red, the down regulation may be shown by the green, and if they are expressing in the appropriate amount, like there is no overexpression or down regulation, then it will be expressed as a yellow. Or we can compare two different sets of the like we can compare two different possibilities where in one case we have treated the cells and in other we have not treated the cells, and then we are in the case where we have treated the cells either with some drug or some molecule of interest, we might want to analyze like whether it is affecting the protein or not. So what we can do is we can perform this microarray analysis. By performing the microarray analysis, what we can do is we can collect the mRNA and then we can check for its expression. So the next question is something based on this. This is the flow diagram which depicts the microarray based whole genome expression technique. Here, the yellow, red, or green circle depicts the level of expression of mRNA in a given sample. When it is very high, it shows the red. When it is down regulated or when it is not very high, then it is green. And when it is equal in both the samples, it is yellow. So here the yellow circle identified, which is marked by the blue arrow, means that the expression level of the given mRNA species is what? So is it equal in both samples A and B? Is exclusively expressed in sample A but not in the B? Is exclusively expressed in sample B? Or is not expressed both in sample A and sample B? So anyone who would like to answer? One. Okay. So correct. The correct answer is option A. It is equal in both samples A and B. And the red, green, and yellow here depicts the overexpression or the underexpression based on the colors. And yellow here shows the that both in both the samples the gene is expressed or the level of expression of the mRNA is similar. The next question is, which one of the following statements is correct with regard to the RNA microarray method of gene expression analysis? Option A, only one color of fluorochrome is used for labeling the RNA species. Option B, the cDNA synthesized from the isolated RNA is labeling with a specific fluorochrome. Option C, the method can be used only for the cancer research. And option D, RNAs extracted from two different sources are directly labeled with two different fluorochrome. So anyone who would like to answer this? Fourth option, I think. Okay, B.
G, ma'am.
Okay. Can anyone explain why it should be B?
Because, ma'am, in case of microarray, they are firstly isolated mRNA and their um, transcriptome was putting in the box of the in the microchip,
Yes.
and then we synthesize the cDNA from the mRNA, and this is the main principle of microarray.
Okay. This is the basic principle of a microarray where we synthesize the cDNA from the isolated RNA and it is labeled with a specific fluorochrome. So the next question is a microarray is a tool which is used to detect the expression of thousands of genes at the same time. Which one of the following options is not correct for this method? That it uses fluorescently tagged samples. It requires the mixing and hybridization of the samples. It requires high throughput data analysis, and it involves the capillary electrophoresis for the for detecting the expression of the genes. Any answers? Okay, Divya has answered option D. Okay. So the correct answer is capillary electrophoresis, as all of the other options, that is the fluorescently tagged samples, are required for the microarray. Mixing and hybridization of the samples takes place for the microarray. High throughput data analysis is required when we want to analyze the microarray data. And capillary electrophoresis is not a part of microarray technique, but it is a part of the northern blotting technique, which is another technique to study the transcription of the mRNAs. So blotting here I have shown the generalized scheme for blotting, which is here it is the southern blotting, which involves the use of the DNA. And when we talk in context of the RNA, it is known as the northern blotting. So the DNA is firstly cleaved with the restriction enzyme and is run in an electrophoresis gel. And based on the molecular weight, the DNA gets separated on the electrophoresis gel. This uh, bands of the DNA are then transferred to the nitrocellulose membrane with the help of a capillary action. So this capillary action helps in the transferring of the bands from the gels to the nitrocellulose membrane. Then we obtain a nitrocellulose membrane that has the marks of the that have the DNA which was there in the gel, and then we can hybridize it with the labeled DNA or the RNA probe, and this labeled DNA or RNA probe can then be visualized based on the what kind of labeling has been done to the DNA or RNA probe. And the autoradiogram generated here shows which what is our gene of interest, or what is our the specific mRNA which is getting expressed or not in the sample. So this is an example of the northern blotting process, where the northern here this one is the uninduced, uh, where we have three different cells, in which one we have which are uninduced, the others are the induced for the differentiation, and we have checked the expression of the beta globin mRNA in differentiated erythroleukemia cells at time points of 48 hours and 96 hours. So we can see that in the uninduced cells, there is no mRNA for the beta globin, and when they were induced for the differentiation, they produce the beta globin at the 48 hours of the time point. Similarly, we can see that the beta globin mRNA is also present at the 96 hours of the after the induction, and the difference in the bandwidth actually tells us what is the level of expression. For example, here we can see that at the 96 hours, the bandwidth which we have obtained is large as compared to what we have obtained in the 48 hours of the time point, and therefore we can say that by 96 hours, there is an increase in the beta globin mRNA in the differentiated erythroleukemia cells. So the next question is based on the northern blotting approach. Northern blotting is one of the functional genomic approach to study the transcriptomics. Identify the option which provides the limitations of the technique. That only one gene can be analyzed at a time. It requires large amount of RNA for the analysis. It is time consuming or all of these. So what are the limitations of the northern blotting technique?
Option D.
Okay. Any other answers? Okay. Divya has also wrote option D. Okay. So the correct answer is option D. All of these are the limitations. As we see that here only one we can hybridize it with a labeled DNA or probe, but this labeled DNA or RNA probe will be against the one specific gene or one specific mRNA. Therefore, at a time we can only detect or only analyze one gene, and it requires a large amount of time and also the large amount of RNA is required because we need to transfer the RNA from the gel to the nitrocellulose or the nylon membrane, and therefore if we use the lesser amount of the RNAs, it may affect the capillary action and thereby we might not be able to detect the mRNAs which we were trying to check. So all of these are the limitations for the northern blotting. Which one of the following option qualifies as a probe for the genome applications? So what kind of probes can be used? The options are
All of the
radio labeled DNA, fluorescently labeled RNA, radio labeled RNA. So all of these can be used as a probe for the hybridization and therefore can be used for the different techniques which we use for the genomics and transcriptomics. So all of these probes, that is the radio labeled DNA, fluorescently labeled RNA, radio labeled RNA can be used. So from the PCR, we can take up in the next class. For today, I would like to end the class here only. If anyone has any doubt, they can. Okay. So can so if anyone has any doubt, they can write in the comment section, and if it is possible, we can take up it in the next class. So is it okay, Ricky, if we'll take it in the next class, as I have to end the meeting?
ma'am, I have also question regarding RNAi.
Okay.
And the
So
question is, suppose we designed an experiment, the suppression of the mRNA.
okay.
Then we entered the complementary gene of this mRNA. Also we are introduced the directly mRNA in the cell from the outside.
It depends. Like there is a difference. miRNA is expressed endogenously, and siRNA, that is also a part of that also leads to the RNA interference. That is what we provide exogenously. So I think it clears your doubt. I you Are you asking this only?
No, ma'am, I actually told that when suppose one cell expressed a mRNA, functional mRNA from the DNA, and this functional mRNA is for suppose cause any disease in our human. Suppose
O-
this is the pathogen genome. Now we treated this disease, use the miRNA technique. Same time we are introduced an complementary RNA regarding to this bacterial or pathogenal mRNA, which he introduced the pathogen in our body.
Okay, right. So
But
this is the natural way how the bacteria actually works, and this is how we have come to the CRISPR-Cas9 approach. So bacteria usually use this CRISPR-Cas9 approach the way you have said, and they use it against the defense mechanism for the various viruses which infect the bacteria. So yes, if we want to interfere, if we want to interfere the expression of the mRNAs of the pathogenic organisms, then we'll need to produce the siRNA or the miRNA against that particular sequence. And Ricky, okay, I'll explain this in this session only. So, what is guide RNA? So, guide RNA is basically the sequence of a RNA which is complementary to the gene of interest, which we want, like which we are in case of CRISPR-Cas9, the gene of interest which we want to like remove or knock out. So guide RNA is complementary to that gene of interest, and basically its composition is same as the RNA, and its role, as I told, that it helps, as the name suggests, that it is a guide RNA. So it guides the Cas9 complex, which is an endonuclease, and that cuts the gene of interest or creates the double strand breaks in the during the process of CRISPR-Cas9. So basically, it guides that complex, and therefore leading to the specific endonuclease activity, creating a double strand break at the specific site, at the site of our gene of interest. Is it clear?
One doubt I have regarding the guide RNA. Guide RNA itself, is it capable of guiding the Cas9 endonuclease to its specific site for
Actually,
action, or it is made up of many components?
basically, this recognizes a PAM specific sequence, and this PAM specific sequence is present near to the this guide RNA. Okay, wait, let me come to that slide. Okay, so this is the PAM specific sequence basically, and this guide RNA recognizes this PAM specific sequence which is present 2 to 3 like which is present downstream to the R cleavage site. And this guide RNA recognizes this PAM specific sequence, and this and therefore guides the Cas9. The way you said that the guide RNA itself is capable. So yes, guide RNA itself is capable of recognizing this PAM sequence, and these actually the Cas9 which we are using and the PAM sequence which we are using is the organism dependent. For example, if I am using Streptococcus, let's say, or so the PAM sequence which is present in those and in that site, and the Cas9 that will cleave the site with the help of the guide RNA at the site, at the PAM site, will be complementary to each other. If you want a better clarification, I can then add up this one more slide in the next session, where different Cas9s are available against the different PAM sequences which are expressed in the different genomes. Is it okay? Hello. Okay. So if there is no other doubt, I'm ending this session. Hope to see you all in the next session.